Okay, let's go ahead and transition to lecture here. Thank you. And now that we've spent some time talking about DNA replication, and ultimately we talked about RNA replication and protein synthesis, now we want to talk a little bit about, well, what happens when something goes wrong? So we'll talk about DNA mutations, and then the fortunate thing for us as living beings here is that fortunately when these things go wrong, we often have ways to correct them. So let's talk about DNA mutation to begin with. And so when we look at DNA mutations, first of all, a DNA mutation is going to be any change that occurs in the sequence of DNA. Okay, so a mutation, again, any change that occurs in the sequence of DNA. And usually when we think about mutations, we think of them as perhaps having some sort of serious detrimental effect on either a cell and or an organism. However, sometimes mutations have no effect at all. And we'll talk about that in just a second here. The DNA mutations, these can be categorized by the type of change that occurs in the DNA. And so this table here shows us a number of these. So for instance, some mutations that occur in the DNA can be what we would call a point mutation. And a point mutation refers to a change that occurs at a single base pair in the DNA. Okay, so again, a point mutation is a single base change that occurs in the DNA. We'll come back to this slide in just a second, but on this next slide here, we see an example of a point mutation where this AGC gets converted to AGT, and then the complementary strand of the DNA is also changed as well. But this would be a point mutation. Okay, so one nucleotide that changes in the DNA. Some terminology that we use here when we talk about point mutations is, first of all, these point mutations can either be considered transitions or transversions. And what I mean by each of these is a transition is when a point mutation, or where a point mutation, let me say that again. So it's when a point mutation is where the change is from a pyrimidine to another pyrimidine, or a purine to another purine. So remember before we said that the A's and the G's were purines, and the C's and the T's, as well as the U's, were pyrimidines. So now, if we're going from a pyrimidine to a pyrimidine, so for instance, a C to a T, right? This one here, we're changing this C to a T here. That would be an example of a transition. Likewise, a purine, an A to a G, right? So a purine to a purine would also be a transition. Whereas a transversion is another type of point mutation, except here it's when a pyrimidine is replaced with a purine, or vice versa. Okay, so a pyrimidine gets replaced by a purine, or a purine gets replaced by a pyrimidine. So with point mutations, we have transitions and we have transversions. In addition, when we think about point mutations, we can also look at these by the effect that they're going to have on the protein that is ultimately synthesized by that piece of DNA. So for instance, some point mutations may be silent. Okay, so when the mutations are silent here, this is when the change in the DNA does not result in a change in the amino acid that is coded for here. So for instance, if we were to look at our genetic code, we would see that CGA and CGG both code for arginine. So even though this CGA, this A here, gets converted to a G, so there's technically a mutation that has occurred in the DNA, but nonetheless, when we look at the amino acid that is coded here, it's the same one. So this is what we call a silent mutation. In addition to silent mutations, we can also have missense point mutations. And missense point mutations are changes that do result in a change of the amino acid that is coded for here. So for instance, if you go back to our CGA, and this, let's say for the sake of argument, that this G gets mutated to a C. Well, now if we were to go back to our genetic code, this CGA would code for arginine. However, now this CCA codes for proline. So this is an example of a missense mutation. And then lastly here, for our point mutation, we can have a nonsense mutation. And this is where an amino acid gets converted to a stop codon. And here's another example. Again, our CGA with arginine gets converted to a stop codon when this C is converted to a U. And you can imagine that if now all of a sudden in the middle of a protein, rather than having an amino acid, we have a stop codon in that protein's mRNA, well, that can lead to a pretty significant change potentially in that protein. So we have point mutations. And this slide here, you can just jot these down, but these are things that I've just mentioned and you should know about. So again, we have silent mutations. We have missense mutations. And we have nonsense mutations. Which of these have the potential to change the structure of a protein? Yeah, so the missense and the nonsense here have the potential to affect a protein and its structure. In addition to point mutations, going back to this table here, in addition to point mutations, there can also be insertions and deletions. So the addition of one or more nucleotides or the deletion of one or more nucleotides. When we talk about insertions and deletions, these may or may not result in what we would call a frame shift in the protein. And this is going to depend upon whether the insertions or deletions occur in a multiple of 3. So here's what I'm getting at with a frame shift. Let's go back to that first. Let me explain what a frame shift is. So a frame shift is going to occur when the reading frame for the mRNA is changed. So let me walk you through this figure here. So on this figure here, this top part here shows very simply a normal DNA sequence. This is just random here, made up, but this shows what it would be normal and what these codons code for. Okay, so you can see the sequence here. And now when we get to the tryptophan, we then see serine, arginine, and histidine coded for here at the end of this sequence. But now, this bottom sequence, again, just a hypothetical example here, let's say there is an insertion here. And now, with this insertion, look at what happens to our codons that is occurring here downstream of the mutation. So these first codons here are all the same, but now with this additional insertion here, our codons are thrown off in this mRNA. And so now, not only is this amino acid changed here to lysine, but now all the other codons that are downstream of this are now off. So now instead of coding for serine, arginine, and histidine, this mRNA with this insertion mutation is now coding for lysine, serine, threonine, and you can imagine how this might be propagated all the way down the rest of that mRNA. So a frame shift mutation is going to change the sequence downstream from that mutation. Okay, and insertions and deletions may result in a frame shift. Yes?
How would it not?
Very good. That was going to be my next question here. How would it not result in a frame shift? Do we have thoughts on that?
By adding.
Oops. Sorry, what's that? Yeah. So we have to add or subtract a multiple of 3. Yeah, exactly. So now, if we were to add or subtract nucleotides in a multiple of 3, so let's just say for the sake of argument here, that we deleted, in this example here, the AGU. That's just, again, for the sake of argument, say we did that. What would follow downstream from where the serine used to be? We would still get arginine and histidine and so forth. So any insertions or deletions in a multiple of 3 is not going to cause a frame shift. Is that clear to everyone here? Okay, so that's an important point to understand here about insertions and deletions.
What if the insertion or deletion are at different locations? Are they specifically at one location, so you insert 3 or whatever it is?
Right, that's a good question. So the question here, I think, would be like, what if you had an insertion of one nucleotide here, but a little bit downstream, you would have 2 more, for instance. Well, right, in that middle piece there, you would have a frame shift, but then after the last 2, you would be back to the original reading frame. So then downstream of that would not be a frame shift.
Typical, or does it usually just happen in the same spot?
It can be. It sort of depends on the extent of mutations that are occurring in a piece of DNA. And so routinely, probably not, but if there is a lot of mutations that are occurring, for instance, if one is exposed to a mutagen, for instance, then that's a possibility for sure. Yep, good question. Other questions?
So you say it does not mutate because multiple of 3?
Say that one more time.
So you say it's frame shift mutation, but it's not mutate?
So let's go back to our definition of a mutation, right? So mutation is any change that occurs in the DNA, right? So we can still have mutations, but these mutations can affect a protein in multiple different ways, right? And so, for instance, we talked about point mutations already, but if we were to talk about insertions and deletions here, these can also each affect a protein in different ways as well, depending upon whether the insertion or a deletion is in a multiple of 3. And the reason why a multiple of 3 is important goes back to the use of codons. And remember, codons are 3 nucleotides that are used to code for a particular amino acid. And so if we were to now insert or delete in a multiple of 3, sure, we add or remove an amino acid per se, perhaps, but that doesn't necessarily change all the rest of the sequences that occur downstream of that, like would happen with a frame shift. Okay, does that answer your question, or did you have something else you were getting at? Okay. How it folds?
Yeah.
Yeah, so the question here is, would that change how a protein folds? And potentially, yes, absolutely. So with a frame shift, absolutely, you're going to change the protein. And we're talking about an insertion or deletion of like 1 codon. Well, it sort of depends where that amino acid would be in a protein. So for instance, if that amino acid is in a crucial, say, alpha helix or beta sheet for a protein, then absolutely, that could affect the structure of a protein. However, if that deletion is, say, let's say there's a flexible loop that goes on for a while and it's not sort of a key part of the structure of the protein, then yeah, you change the structure of the protein a bit, but it's not really going to impact the protein that much. So it just sort of depends on what the change is and where that would be in a protein. So it's a good question. Other questions? So on one hand, we can look at changes in the DNA and ultimately the proteins based upon what we've been talking about. In addition, we can also look at mutations as whether they are spontaneous or induced. And so we'll talk about induced mutations in a minute, but for spontaneous mutations, these are going to be mutations that occur. So they're mutations that occur sporadically in a cell's DNA without the addition of a reagent. Okay, so again, for spontaneous mutations, these are mutations that occur spontaneously in the cell's DNA without the addition of a reagent.
Spontaneously or sporadically? Or both?
That actually both. Yeah. So they will occur spontaneously and sporadically. Thank you. I didn't realize I had switched those words, but yes, both would apply here. These spontaneous mutations often occur during certain situations. So for instance, they can sometimes occur during DNA replication. We like to think of DNA replication as being perfect and 100% accurate, but as it turns out, there is a low mutation rate that is associated with this. And so it's possible that just during regular DNA replication, that the DNA polymerase may get things wrong. And in this example here, it's showing a point mutation being formed. So rather than having this TA found here in the DNA, during the DNA replication, perhaps a C is added across from the A instead. Oftentimes this gets repaired, but sometimes it doesn't. And so sometimes, again, just through the normal DNA replication process, a wrong nucleotide gets added. And this would be an example of a spontaneous mutation. Another example is shown here. And remember, back when we were talking about DNA, I brought up the idea of tautomers and how the nucleotides, how the bases in the nucleotides can undergo spontaneous changes, spontaneous shifts in their their structure. Right? So for instance, with a thymine here, you can see that this double bond here with this oxygen, you know, occasionally this thymine can undergo a tautomeric shift that results in the double bond being found here. And so each of the bases have the possibility of undergoing this spontaneous tautomeric shift. Normally, right, the nucleotides on the left here are the ones that are most often found in the DNA by far. However, what happens now is if just before DNA replication occurs, there is a tautomeric shift for a base that occurs, well, what happens now is that polymerase, that DNA polymerase, is going to insert the wrong nucleotide opposite that, that base that has undergone a tautomeric shift. So for instance, with thymine, right, what normally is opposite of thymine? What's complementary of thymine? Adenine, right? But now, when the thymine is in its tautomeric form and DNA's replication is taking place, instead of an adenine nucleotide being added, a guanine nucleotide is added instead. Okay, so that would be an example of a spontaneous mutation. In addition, likewise, for adenine, when that undergoes a tautomeric shift, and if that tautomer is found during DNA replication, the DNA polymerase would add a C across from the A instead of a T. And likewise with guanine, if guanine is found in its tautomeric form, instead of a C being added, we would have a T being added instead. And so you can see here how this can lead to a spontaneous mutation in the DNA. For the cytosine, this is the one piece of good news here. For the cytosine, both the normal and the tautomeric form both will base pair with guanine. So for cytosine, it doesn't matter here. We get guanine opposite no matter which form of cytosine is present. But nonetheless, for these others, when they are in their tautomeric form and DNA replication is occurring, we can get the wrong nucleotide being added. And then lastly, a third example of a spontaneous DNA mutation can occur with what we call replication slippage. And what happens here is this takes place when a stretch of DNA is being replicated, and the stretch of DNA has short repeated sequences. And so you can see in this example here that we have the short repeated sequence of CA, 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 and so forth. Well, what can happen here is occasionally the DNA polymerase, if I were to personify it here, I would say that the DNA polymerase gets confused for just a second. Okay, obviously it doesn't get confused, but nonetheless, what happens here is the DNA polymerase, as it's synthesizing this repeat sequence, it can slip upstream or downstream just a little bit. And in doing so, in this example here, it slips slightly upstream, and in doing so, it essentially resynthesizes a small stretch of that DNA, resulting in an insertion or potentially a deletion, depending on which direction it is slipping. This example here shows that we end up with an addition of a CA And now we have in the DNA, we have an extra CA that has been added. Would this be an example? So if we add a CA here, would this potentially be an example of a frame shift mutation? I hear yes and no. So the answer is this is potentially a frame shift mutation. And the reason is because it's not in a multiple of 3, right? So we're adding just a C and an A here. We're not adding a third nucleotide in this example. So this has the potential to cause a frame shift. Okay, so for spontaneous mutations, we have one, it normally occurs during DNA replication. We have 2, the tautomers can increase that rate of spontaneous mutations. And then 3, replication slippage is another way by which spontaneous mutations take place. Replication slippage can actually relate to a particular disease. Have you talked about Huntington disease yet in Pathophys? If not, you will soon. And Huntington disease is a neurodegenerative disease. It can lead to the loss of various cognitive facilities, chorea, so uncontrolled writhing movements. Patients decline eventually, so they can no longer take care of themselves. With Huntington disease, there's variable age onset, but it often strikes during the Middle Ages. And the reason why I bring this up here is because it relates to an insertion that occurs in the Huntington gene. And the Huntington gene codes for a protein that seems to play a role in causing Huntington disease. And what happens here is in the normal form of this Huntington protein, or in the normal form of the Huntington gene, I should say here, there are about 6 to 31 CAG repeats that are found here. Okay, so 6 to 31 of these CAG repeats. However, what happens here is for individuals who experience Huntington disease, these individuals have a greater number of these CAG repeats. Okay, so somewhere along the line here, for whatever reason, these individuals now have additional insertions in this gene. Okay, so they have greater CAG repeats, and as it turns out, there's a correlation between the more CAG repeats that one has and the onset and severity of the disease. And so what happens here is these mutated Huntington proteins, they aggregate in neurons and can lead to the cell death of the neurons. So these insertions and deletions here, here's an insertion, and other mutations can have some pretty dire consequences for a cell as well as an entire organism. Okay. So next class, what we'll do is, now that we've talked about spontaneous mutations, we'll then talk about induced mutations and how mutations can be triggered in DNA. But for today, we will leave things off here. If you have any questions, feel free to come up and ask them. Otherwise, we're finished, and we'll see you on Friday. Thank you.
It said A frame shift. Now, is that A like the vocabulary A, or like any frame shift? If it was a G frame shift, would it